Good morning students. I am Dr. Abhishek Kumar Singh, Tutor, Department of Physiology, and I am continuing with the new chapter in the CNS about which we talked about in the last class. That was Introduction to Sensory System. We had talked about this in the last class. We'll just give a quick revision to that. First, sensation, that is basic recognition of stimulus. Then absence of sensation is called anesthesia. Abnormal sensation is called paresthesia. And sensation of movement is kinesthesia. Then the types of sensations we talked about: special, visceral and somatic. And then the classification of sensation, that is general sensation and special sensation. The general included superficial, deep superficial and deep sensations, and the special sensation included vision, audition, taste, smell and linear and rotational accelerations. Then we also talked in detail about dimensions of sensation under 4 categories mainly, that is modality, intensity, effect and acuity. So today we are going to start the second part of this chapter, that is Introduction of Sensory System and Physiology of Receptors. So today we are going to talk in detail about the physiology of receptors. First, what are receptors? Receptors are transducers that convert various forms of energy in the environment into the action potentials in the sensory neurons. So they are converting the energy into the action potential in sensory neurons. Now what are their properties? First, they are the endings of afferent nerve fibers. These may be a part of neuron or specialized cells that generate an action potential in the neuron. They are usually associated with non-neural cells that actually surrounds it. For example, receptor plus the non-neural structure forms what is called as the sense organs. And then third thing about receptors, they are specific for a particular stimulus. The form of energy to which the receptor is most sensitive is called adequate stimulus. Example, light for rods and cones of retina. You are already going through this chapter in eye. Then these are the properties of receptors. After knowing the definition of receptors, the properties of receptor, we come to classification of receptors. How are they classified? The classification is based on their functions, based on their adequate stimulus, based on their location. First classification, which is based on function, which is you are seeing projected in the slide here. Based on function, they can be exteroceptors, they can be interoceptors, they can be proprioceptors, they can be teleceptors. Now, 2 lines about each of these. First, exteroceptors, for example, skin and subcutaneous tissue, which are concerned with changes in the external environment close to the body, and they are present in cutaneous sense organs. They are exteroceptors. For example, you can see here temperature receptors and pressure receptors in the skin. Location of stimuluses, they are immediately from outside the body. Second is interoceptors. Interoceptors detect changes in the internal environment of the body. Examples are baroreceptors for BP changes, which you will study in cerebrocardiovascular system. Third is proprioceptors. Proprioceptors are the receptors which provide information about the position of the body in the space at any given time. So this is providing the position of the body. Examples are stretch receptors in muscles and balance receptors in ear. The sense of position is called as kinesthesia, which we have already seen when we are talking about introduction of sensory system. Then telereceptors. Telereceptors are the receptors which receive stimulus present far away from the body. Exteroreceptors receive stimulus which was present close to the body, while the telereceptors are those which receive stimulus present far away from the body. Example, auditory receptors which detect sounds from the distance. So it receives stimulus from a distance outside the body. That is the location. An example can be auditory receptors or olfactory receptors or the cells of the retina. They receive stimulus from the distance outside the body. They are called telereceptors. So you have to know why they are called external why they are called interoreceptors, proprioreceptors and telereceptors. So they are based on their function. Second classification, or the classifying the receptors based on their mode of activation, or you can even say based on their adequate stimulus. What are the various stimulus which we see, like touch, pressure, cold, warm, pain, chemical stimulus and photon stimulus. All these are detected by various sense organs in the skin as well as in the subcutaneous tissues and the epithelium. The important receptors are mechanoreceptors, thermoreceptors, nociceptors, photoreceptors, chemoreceptors, osmoreceptors. Mechanoreceptors are those which responds to application of a mechanical stimulus, that is, they are sensitive to deformation. Example are either touching or stroking of skin. Now these mechanoreceptors can be divided into three categories. There can be expanded endings, there can be encapsulated endings and there can be naked nerve endings. Example of expanded endings of mechanoreceptors are Merkel disc and Ruffini's ending. Examples of encapsulated endings are Pacinian corpuscles, Meissner's corpuscles and Cross's end bulbs. And the naked nerve endings are those where all the four cutaneous sensory modalities can be elicited from a skin of the area which contains only naked nerve endings. So mechanoreceptors have further three categories and we'll talk in detail about mechanoreceptors in the next few slides. Then what are thermoreceptors? As the name suggests, which detect changes in temperature. Then there are nociceptors which responds to painful stimuli. These nociceptors are of two types. A delta, which are mechanical nociceptors which responds to sharp pain, and then there are C polymodal nociceptors which responds to several types of noxious stimuli, which includes either thermal and chemical. Thermoreceptors which detect changes in temperatures are again of two types. They can be warmth receptors or cold receptors, and they are active over a broad range of temperature changes. In details you can study in the textbook. Then chemoreceptors are those which responds to chemical stimuli like taste receptors and olfactory. Then photoreceptors are those which are sensitive or activated by the photons of light. Example, rods and cones of retina. Then osmoreceptors are those which detect the osmotic pressures of body fluids. So these are based on their mode of activation, or you can say the classification is based on the adequate stimuli. Then the third way of classifying them is based on their location. They can be superficial like touch and pressure receptors. They can be deep, those are the receptors present in muscles, bones and tendons, etc. Then there can be visceral, which are present as visceral pain receptors and on the visceral organs. Now we come, and even there is one more way of classifying the receptor, that is based on their complexity. There can be simple receptors and there can be complex receptors. And what are those complex receptors? The sense organs are the complex receptors, about which you have already been studied, all the sense organs. Then we come to what are the various important mechanoreceptors. We talked about mechanoreceptors, three categories: the expanded endings, the encapsulated endings and naked nerve endings. The first and the one which is the largest mechanoreceptor is the Pacinian corpuscles, and you can see the structure, the diagram of this Pacinian corpuscle, how it looks like. It was discovered by Filippo Pacini. The name from that is derived as Pacinian corpuscles. Where are they located? They are located in skin and deep tissues, and they mediate the sensation of vibration, touch and pressure. Each of these lines can be an MCQ question. The structure, you see the structure, the diagram shown here. It consists of a body which is composed of concentric layers. These concentric layers are, you can see these concentric layers. These are called lamellae. These concentric layers are the lamellae. They look like the pills of onions, and there can be 20 to 70 lamellae in each corpuscles, which are arranged. How are these arranged? They are arranged concentrically. They are concentrically arranged, these lamellae. Now, what is the dimension? They are the largest mechanoreceptors. The dimension is 0.5 to 2 millimeter long. The distal end of a primary efferent fibers penetrate into the concentric lamellae of the Pacinian corpuscles. This is what the distal end is penetrating. This distal end is penetrating this concentric lamellae, which you can see here in the diagram. Now the fiber is myelinated. This is myelinated, as you can see the Schwann cells here. The fiber is myelinated, but the ending in concentric lamellae, this is the endings in the concentric lamellae, this is unmyelinated. What is their function of this Pacinian corpuscles? They are rapidly adapting mechanoreceptors which are capable of receiving high frequency vibrations, and therefore it senses vibration, fine touch, and also pressure sensations. The distal end of the axon, which is the distal end of the axon, not the lamellae, is the actual transducer. So this is not this lamellae is not the transducer, but the distal end of the axon is the actual transducer. Then we come to another important type of mechanoreceptor, which is called as Meissner's corpuscles. You can see in the diagram here, this is the Meissner's corpuscle. They are relatively large, located in the dermal ridges of hairless skin. They are encapsulated endings having a single layered capsule into which the endings of afferent nerve penetrates. You can see this. The endings is branched to form complex structure inside the corpuscles. These endings of Meissner corpuscles are branched inside to form a complex structure. What is their function? They are rapidly adapting receptors that senses vibration sensation and also sensation of contact, that is touch. Then another type, which you can see the name here, Merkel's disc. Merkel disc is the most superficial mechanoreceptors present in the dermis of glabrous, that is the hairless, as well as in the hairy skin. And what is their function? They are slowly adapting, that senses touch and pressure. You can see here, this is the Merkel's disc. This is the Merkel's disc. This is the diagram of the Merkel's disc. And then we have Ruffini's ending. This is Ruffini's ending. This one. This is the Ruffini's ending or Ruffini's corpuscle. They are the smallest mechanoreceptors present in the hairless as well as in the hairy skin, and their function is slowly adapting receptors which receive sensations of crude touch. We have fine touch, we have crude touch. So Ruffini's corpuscles, which are the smallest mechanoreceptors, Meissner's Pacinian corpuscles, which are the largest mechanoreceptors. You can get them as an MCQ questions. Then there is one more, which is called as the Cross's end bulb. They function as mediators of sensation of touch as well as pressure, and they are again rapidly adapting, present in dermis and widely distributed in skin. The name is Cross's end bulb. Then we come to receptor potential or generator potential. On application of, you can see in the diagram is very much self-explanatory. On application of a stimulus, potential change is observed in the receptor, which is non-propagated depolarizing potential that resembles what we already studied, excitatory postsynaptic potential, and this is called as receptor potential or generator potential. I'll just repeat. When a stimulus is applied, potential change is observed in the receptor, which is a non-propagated depolarizing potential which resembles EPSP, and this potential is called as receptor potential or generator potential. How is this? These are non-propagatory types, 1, 2, 3, and then the 4 propagates. This one 4 and then 5, it propagates. So this is undergoing summation, and thus the excitatory postsynaptic potential, which is the receptor potential or the generator potential. How it is generated? What is the mechanism of genesis? Stimulus of some intensity is applied, then the magnitude of the potential increases, which reaches about 10 to 15 millivolt, which results in action potential in the sensory nerve. Here, the intensity of the stimulus will be directly proportional to the magnitude of potential produced. The lamellae of the Pacinian corpuscles not required for receptor potential. It is if the unmyelinated nerve terminate, then generate the receptor potential. So we are taking the example of Pacinian corpuscles in explaining the receptor potential to you. Now where this is generated? This is actually generated at the first node of Ranvier, where the action potential is generated, and the magnitude of action potential will directly depend on the intensity of the stimulus. Then we come to the third and the last portion of the chapter, which includes the properties of receptors. What are the various properties which a receptor owns? First is specificity. As the name itself suggests, it is specific to a particular stimulus. One receptor is specific to one particular stimulus. That is our first property. Second property is adequate stimulus. Means a particular form of energy to which the receptor is most sensitive is called as adequate stimulus. The adequate stimulus for rods and cones is the photon, that is the light. Third is adaptation. Means the stimulus of a constant strength when applied continuously to the receptor, that causes the frequency of the action potential in the sensory nerve decrease, which results in adaptation, also called as desensitization. When a stimulus is constantly applied, that decreases the frequency of action potential in the sensory neuron, and that is called as desensitization or adaptation. Based on this, receptors can be phasic receptors and tonic receptors. Phasic is one which adapts rapidly, like the touch and pressure receptors, and tonic receptors which adapt slowly. Example, baroreceptors and pain receptors. Sometimes pain receptors do not adapt at all. Then the fourth property of receptor is acuity. Acuity means precision of stimulus localization, which depends on the number of stimulus present in that area where the stimulus is applied. Fifth is intensity. The strength of the stimulus is the intensity of the stimulus. So when a strength is more, the activated neuron fire at more rate, and the receptor present at some distance away can also receive the stimulus, and that is called as, and also discharge, and that is called as receptor recruitment. So the term comes here as receptor recruitment, which means when the strength is more, the activated neuron fire at more rate, and the receptor which are present even at some distance away from the stimulus may also discharge, and that is called as receptor recruitment. Then there is something called as Weber Fechner's law. The Weber-Fechner's law includes, or it actually explains, that the magnitude of sensation felt is directly proportional to the log of the intensity of the stimulus. The magnitude of sensation felt is directly proportional to the log of the intensity of the stimulus, and this is the Weber-Fechner's law. You can even go in detail about this Weber-Fechner's law to make you understand more about it. Then the seventh property of receptor is law of projection, which means sensation formed is referred to the location of the receptor. This forms the basis of phantom limb. Means even when a person has an amputated knee, amputated leg, he may get a sensation of pain in that amputated leg which is no more there, and this is the basis of the phantom limb. So that is due to this law of projection. Then I'll just talk in one line about the Mueller doctrine of specific nerve energy. What does this mean? It is not shown in the slide here, but I'll just give you one short line about this, that when a sensation is evoked, the sensation is formed, it evokes a stimulus that generates an impulse, and in a specific sensory pathway it depends upon the precise area of the brain that is activated by the stimulus, and that is called as the Mueller doctrine of specific nerve energy. That's all about the properties of receptors and the chapter sensory, introduction to sensory system and the physiology of receptors. In the next class, we'll talk a new chapter in CNS, which will be the sensory communication to spinal cord. Thank you.